So, good morning, ladies and gentlemen. It's a great pleasure and honour to be here giving the opening lecture for this wonderful Insigneo Showcase day. And as Marco has said, I'm here really in my capacity as Professor of Neurology and Director of the BRC. Marco asked me to tell you a little bit about the Sheffield BRC, which has just come to the end of its first year of the 5-year funding. So that's what I'm going to do in the next 20 minutes or so. And what we're trying to do with the BRC is to harness all the exciting developments in science, neuroscience, in silico medicine and so on, harness those exciting discoveries and translate them into new cutting-edge treatments, technologies, diagnostics, interventions for patients with neurological conditions. So the BRC sits between all the important basic science happening in multiple institutes that have joined forces, as Marco said, to create the BRC. The BRC enables us to pull through those discoveries into experimental medicine, early phase trials, looking for biomarkers, that sort of thing, to pull those into studies in human beings, which take place in our other partner, Sheffield Teaching Hospitals, and then if successful, those experimental medicine studies will go through into later phase clinical trials to prove efficacy and cost effectiveness. So I think the interjection or the bridging of the Sheffield BRC enables us to really follow through this therapy discovery pathway. So very exciting. And as Marco said, we pulled together talented individuals with a huge amount of goodwill. So we joined forces in a very powerful way to create the BRC. So the Insigneo Institute, the Sheffield Institute for Translational Neuroscience, Sheffield Teaching Hospitals, colleagues in the Faculty of Science as well, all joined forces together. The structure of the BRC, I don't think I need to dwell on this, so I'm currently the director. Chris McDermott is the deputy director. We have a very able manager, Jodie Keyworth, who has a lot of experience in human studies. She was the research coordinator in the Academic Directorate of Neuroscience at STH. Oliver Bandman's our training lead. We've brought through a whole raft of clinical fellows and PhD students, and we have a team now assembled to really facilitate those experimental medicine studies that we're aiming for. And what we're working on, if you like, is depicted here. So we were advised to be, we started out initially with a broader-based BRC, but NIHR advised us to focus down on neurological disorders, so that's what we did. So our 3 pillars are neurodegenerative diseases, motor neuron disease, which is my baby, of course, Parkinson's disease, dementia and the aging brain, neuroinflammation, especially multiple sclerosis, but disorders of the cerebellum, ataxia as well, and then the important pillar of cerebrovascular disease. And supporting those healthcare themes are our cross-cutting themes, advanced medical imaging, genomics and bioinformatics, the clinical research facility supported by NIHR in the hospital, but most importantly from today's point of view, in silico medicine with Marco and Claudia Maza leading the Insigneo input into the BRC. We think, I would of course as a neurologist, but we think the disorders we're focusing on are very important for the population in Europe and worldwide. So 1 in 3 of us in this room will develop one of the top 3 neurodegenerative disorders in our lifetime. And as we're no longer dying of tuberculosis and diseases like that, we're living to a riper old age, and these diseases are increasing exponentially. Multiple sclerosis is the commonest cause of disability in young adults in the UK, so a very important disorder. And stroke, of course, a really important leading cause of disability and indeed death. So what we're trying to do in relation to these healthcare pillars is try to establish standardized ways of observing patient symptoms and the underlying biology of their diseases. We want desperately to identify biomarkers of disease progression and, most importantly, response to treatment. We want to subclassify the diseases better, so categorize patients with similar biological characteristics into groups so that, as the elegant film showed at the beginning, so that we can do precision medicine and tailor treatments and therapies to the right groups of patients. And we want to use all this information to actually do better for our patients. So I think neurological disorders are a bit like infectious diseases was a good number of decades ago. We're teetering on the brink of being able to protect neurons better, but we're not there yet, and there's still a lot to be done. So I'm just going to give you some highlights of the sort of thing that we're doing. So let's think about stroke first. So Arshad Majid is our Professor of Stroke Neurology. He leads the stroke sub-theme, and they're doing lots of studies aiming to get better rehabilitation outcomes after somebody has had a stroke. So they're looking at remote ischemic conditioning to improve post-stroke fatigue, which is a major problem. They're also looking at the effect of this remote ischemic conditioning on cerebral perfusion. They're using a very elegant, non-invasive vagal nerve stimulation, which you can get at a little branch of the vagus nerve just behind the ear and non-invasively stimulate it at that site. And there is promising evidence that using that improves the outcome in somebody with a disabled upper limb following a stroke. So it can improve the recovery of upper limb function. We've got a whole pipeline from the work of CITRAN, the Sheffield Institute for Translational Neuroscience. We've got a whole pipeline of therapies coming through from the basic science work in CITRAN that are at various stages of coming through into human trials. I won't go into all of those in detail, but we've got neuroprotective drugs for Parkinson's disease, neuroprotective drugs for motor neuron disease, gene therapies, approaches for various neurodegenerative disorders, new treatments for the prevention of major stroke. And you'll see from this busy slide that we're at various points along the pathway to proof of concept in human studies, but that pharmaceutical and biotech partners have joined forces with us in a major way to pull through those scientific insights into translational benefits, we hope, for human beings. So let me talk a little bit about MND. I got interested in it as a young neurologist just because it made me feel like a useless doctor, and I thought it was just about the worst disease in medicine. So if you ask a room full of doctors what disease they would least like to develop in their lifetime, many of them would say motor neuron disease. So in MND, we're doing quite a lot of experimental medicine studies now, phase 1 and phase 2 clinical trials through the BRC. We've got funding for systematic biosample collection, so we can collect blood, urine, cerebrospinal fluid, DNA, RNA, skin cells, fibroblasts, which we can now reprogram into the CNS cells that go wrong in the disease, so we can make them into neurons and astrocytes in the laboratory. But while we're waiting to try and develop better neuroprotective therapies to slow down the course of MND, which usually kills people within about 2 years of the first symptom, there's a lot that can be done to improve quality of life and life expectancy by just paying attention to the troublesome symptoms that the patients develop, and I'll show you an example of that in a minute. So gene therapy, very exciting from the basic work of Citran. So spinal muscular atrophy, SMA, is a childhood form of motor neuron disease caused by a deficiency of a particular protein called SMN. You can, by replacing, using gene therapy to replace SMN, you can cure mouse models of SMA. And that gene therapy approach has been applied to human patients in the US from Ohio, and children that would have died in the first 2 years of life from SMA are now living, running around, reaching their motor milestones. So really, really exciting. A similar approach for familial motor neuron disease. The first gene that was found is called SOD1, SOD1, not a bad name for a gene that causes the worst disease in medicine. But by knocking down the expression of that rogue gene, again, you can make a dramatic difference to the life expectancy of the mouse models. We injected the viral vector into the CSF of the mouse, and we're bringing those studies now through. Just all the safety and toxicology studies are being done, and we'll bring that through into human studies. But in the meantime, we're working with Biogen on a gene therapy approach. So this is using antisense oligonucleotides to knock down the expression of the rogue gene that causes one type of MND. The medicine has to be given into the spinal fluid, so injections into the CSF every 3 months, but really exciting. So patients have been sending videos of themselves walking up the garden steps, which they hadn't been able to do for 3 years before they started the treatment. So we're very optimistic that for one subtype of MND that might work. Ambrosia is just the longitudinal biosample collection. That study is funded by the MND Association. It's taking all those biosamples that I talked about, but each patient is genetically subtyped, so we know of 30 genes now that can cause MND. We have blood markers, urine analysis, CSF markers, careful serial clinical observations, and those samples are collected longitudinally. So that's a really nice model for one disease that we can roll out to the other diseases that I've talked about. Again, from the scientific work of Citran, we pulled out this pathway, NRF2, the NRF2 antioxidant response pathway, as a promising neuroprotective approach. So NRF2 is a transcription factor which, when a cell is under stress, it goes into the nucleus and upregulates a whole lot of cytoprotective genes. And there's promising evidence that that may be helpful in both MND and Parkinson's disease. We've done a lot of work on that. We've got orphan drug designation for one of the top hit drugs that we identified in Citran. There's now an American company helping us take that through to the clinic. And through Richard Mead, we're also, we formed a spin-out company, Keepstone Therapeutics, that is designing a novel way of upregulating that programmed cell life transcription factor, NRF2. and we're working with Parkinsons UK to take some promising molecules through the medicinal chemistry that needs to be done to get a molecule that gets into the nervous system. Tom Jenkins is working with imaging colleagues. Professor Wilkinson here in the audience has been looking at muscle imaging using MRI, and you can actually see evidence of muscles losing their motor nerve supply through T2 imaging changes that happen in muscles. And Tom and Ian have been able to show that with disease progression in a neuromuscular disorder like MND, you can see changes happening to that muscle imaging biomarker. I mentioned that there's a lot we can do about troublesome symptoms, and this is a really nice example of where we worked with engineers and people from Insignio. So in a lot of neurological disorders, the neck muscles become weak, so the patient can't hold their head up properly, chin down on the chest. If you try that for a few minutes, it's really uncomfortable. It means you can't eat properly, you can't look people in the eye when you're talking to them. So a nasty, uncomfortable problem, and it's painful. Your head is surprisingly heavy. It weighs about 5 kilograms. So, and Chris McDermott from the BRC led this study. It was a lovely study, really, with real engagement from patients and the public. So patients, family members told us what was wrong with the existing neck supports and what they wanted from a new one. So it was an amazing team: neurologists, nurses, physiotherapists, patients, bioengineers, material scientists, even fashion designers. And so this is engineering meeting neuroscience. They had a very fancy way of measuring the pressures and forces on the neck, and all of this elegant teamwork ended up producing this head-up collar that then patients tested, and the design was refined a little bit according to their feedback, and it's cosmetically very acceptable compared to the clunky existing collars, and it's now tailor-made. Have taken it to market. Patients all around the world have been waiting for this, and it's now available for them. And the beauty of it is it's adjustable. So if one side of your neck is worse than the other, you can bolster one side, you can have extra support when you're bouncing along, driving on some of the potholes in the Sheffield roads. So that's a very nice example of where you can just improve quality of life by paying attention to troublesome symptoms. Dementia, I'll just mention briefly. Anna-Lena Veneri is our Professor of Translational Neuropsychology, working with colleagues in neurology. They've just published a very nice biomarker study showing that the volume of a particular area of the brain known as the ventral tegmental area may be a very good marker of who is going to develop Alzheimer's disease in a patient with apparent mild cognitive impairment. Oliver Bandman and his team really working with skin cells from patients, zebrafish and fruit fly models, identified that the batteries of the cells, the mitochondria, are not functioning properly to generate ATP in Parkinson's disease. Having established that in animal models and human cells, they then screened drug libraries to see what drugs might correct that defect. And they came up with ursodeoxycholic acid. And Oliver has now obtained funding to take that drug. It's a repurposing because that drug is already in human use for other conditions, but they're now taking it into a phase 2 trial and working with Insignio colleagues. So here's Claudia Marza. They've nested into that experimental medicine study some very elegant new design aspects. So they're objectively quantifying motor activity, they're using imaging to be able to quantify the amount of ATP produced by mitochondria in the human brain. So very exciting. There's a big collaboration. So Claudia is our movement analysis guru in the BRC. She works with colleagues in multiple disease areas, including Basil Sharrock, who leads the MS clinical service in Sheffield. Really, Basil and his team have come up with a very exciting breakthrough. I think you would call it a breakthrough in MS. So patients with aggressive, relapsing, remitting disease, if you extract bone marrow, take the stem cells out of the patient's own bone marrow, and then immunosuppress them, so ablate the existing bone marrow, put those stem cells back into the same patient, you seem to reset the immune system and stop the disease in its tracks. And this MIST trial, the results have just been presented, they're not yet published, but a really dramatic improvement in outcome for patients who had that autologous bone marrow transplantation. Panorama produced a program on it. It's been presented at the European Neurological Association as a major breakthrough, and they've now got NIHR funding to take that further, to now compare in a controlled way best existing disease-modifying therapy against bone marrow transplants. So really exciting. In the last couple of minutes, I'm going to just talk a bit more about the cross-cutting themes. In the interests of time, I can't cover all of them, but I'll just show you one or two highlights. So Ian Wilkinson, sitting back there, leads the advanced medical imaging theme. The president of the day, the leader of today, Marco, leads the in silico medicine cross-cutting theme. Winston Hyde leads the genomics and bioinformatics theme, and Chris Newman, who directs the CRF, leads the clinical research facility cross-cutting theme. So let me, because this is about Insigneo, let me just talk about some of the things that Insigneo is contributing to this neuroscience BRC. So what the Insigneo team are trying to do really is help us develop biomarkers for progression in movement disorders. So by gait analysis, muscle strength assessment, remote activity monitoring with wearable sensors. We want to integrate, later on, we want to integrate movement data with imaging data. And in the longer term, we want to be able to test predictive patient models for in silico augmented clinical trials, really, to compare with experimental medicine results. So that's what we're aiming to do. The gait analysis, so Alistair McNeil is an Insigneo fellow. He's been using this sophisticated gait analysis system, OptoGait, which detects footfall with spatiotemporal sensitivity. So you can see the footprints there. You can measure at the same time how much the upper body is swaying when the patient is walking. The Insigneo team have also developed MATLAB scripts to be able to analyze data from the sensors that are used for this. So just as an example, in patients with unsteadiness due to cerebellar ataxia, you can detect using these systems that the affected patients have a shorter and wider gait. So if you compare the footprints from normal people versus cerebellar ataxia, you can see the broad-based gait and the shorter steps. But importantly, you can also find changes in pre-symptomatic individuals, so an early way of detecting motor system problems. And they've used this system in a condition called DiGeorge syndrome. That patients with that syndrome have a 20-fold increased risk of developing Parkinson's disease, and they're using that system to predict who is at risk of developing Parkinson's disease, so they can detect reduced speed of gait, reduced cadence at an early stage. So exciting ways of detecting medical conditions, neurological conditions very early on. We worked with STH, who very generously found us some space to put the staff of the BRC, and we incorporated into that a biomechanics lab. So we can actually analyze Claudia and her team, her new PhD student, Lorenza, can take neurological patients into this facility within the hospital and do all their fancy motor analysis there. Just a little word in my last minute or two about imaging. So I think we have real strengths in imaging in Sheffield, and I know many of the imagers work with Insignio very closely outside the nervous system. You may find that a bit deranged to be interested in systems outside the nervous system, but in relation to the cardiovascular system, musculoskeletal system, imaging is a real strength, I think, both in the university, in my faculty, in the Faculty of Medicine, Dentistry and Health, but also in engineering and Insignio. And the work that Jim Wild leads using really innovative approaches, hyperpolarized gas, to image the lungs. And I'm delighted to say he's pushing that towards the nervous system as well. So we've got super imaging teams, and as part of the BRC, we're actually enhancing the infrastructure for imaging. So we're going to set up a combined PET MRI imaging system. So that combines the detailed anatomy that you can get from MRI with the functional readouts that you can get from injecting PET tracers. So really useful for neurological disorders, but many other disorders as well. So oncology, diabetes, for example. So we're working hard to get that set up. We hoped we would be able to slip it into the radiology facility in the Hallamshire Hospital, but we actually found we have to produce a new building, so it's been a little bit more complicated than anticipated. But Marco talked before about goodwill and joining forces, and I think developing this £10 million PET MRI facility is a real example of that. So the power of people, the power of the goodwill of people. So starting with Sir Keith Burnett and Sir Andrew Cash, who really got behind this, all sorts of skilled people in imaging. Carl Herholtz from Manchester was our advisor on PET imaging. The fundraising team under Miles Stevenson in Dare, and the estates departments from both the university and the hospital working together to make this happen. This is where it's going to be, so it will join the existing academic radiology unit. We've just got planning permission from Sheffield City Council, and the builders will be on site in the next 4 to 5 weeks. So we're expecting by this time next year it will be up and running, and we look forward to doing a lot more work with Insignio colleagues in relation to this molecular imaging that we'll now be able to do. So I'm going to end there. I've skipped you through a lot of things, but I hope it's given you a flavour of what the BRC is aiming to do. And as Marco said, we've got a foot in the door in relation to neuroscience, but it shows what we can do when we join forces. And I think one of the special things about the University of Sheffield is that we are a friendly place, and it's not difficult to collaborate across the disciplines. So medicine with engineering, with basic science, and so on works extremely well. So thank you for your attention. I'm not sure I've allowed time left for questions, but I'm happy to answer any if you have questions. Thank you.